Okay, madam, you can start.
Okay, sir. Okay, a very good afternoon to all of you. I welcome you, Dr. Sorish Bhattacharya, for presenting a webinar on a very interesting topic, that is the valorization of microalgal biomass for development of nutraceuticals, biofuel, and recovery of bioplastics. Let me briefly introduce Dr. Bhattacharya. Dr. Sorish Bhattacharya has completed his BTech in Biotechnology from West Bengal University of Technology and MTech in Fermentation Technology from Institute of Chemical Technology, Mumbai. And he did his PhD from ACSIR, CSMCRI, and presently he is working as a senior scientist at CSIR, Central Salt and Marine Chemicals Research Institute, India. He is having a strong background in the area of biochemical engineering, microalgal biotechnology, including microalgal biofuel, biopolymers, and nutraceuticals for therapeutic application. He have published 26 papers and have 12 book chapters and 3 patents to his credit. Now I welcome Dr. Sorish Bhattacharya, and yeah, I request you to please start.
Yeah, thank you, ma'am, for a wonderful introduction. Today, we would be discussing on the valorization of the microalgal biomass for developing various nutraceuticals, biofuels, and recovery of the bioplastics for value additions. We all know that nowadays, considering the fact that the crude oil prices have increased to a very large extent, so in this context, all the lignocellulosic biomass and other feedstocks are currently not having a sustainable approach. So in that area, we all have known about the biofuel policy as well, wherein 10 to 15% blending needs to be done, but still there are certain issues. So what we are thinking is let's have a biorefinery approach wherein microalgal biomass can be used as a feedstock. So coming into the question that why we think of microalgae. So there are varieties of problems that can be addressed through the microalgae. That is, it can very well use flue gases which containing carbon dioxide, NOx, etc. and we can utilize it in the growing the algae. Simultaneously, various wastewater can be utilized in a very cost effective manner, and it can provide us a variety of products starting from animal feeds, algal biofuels, fertilizers, nutraceuticals, carotenoids, etc. So then comes the upstream process. So basically there are various strategies to grow the microalgae in open ponds as well as raceway ponds are used for producing nutraceuticals. Some of the people are also using vertical bags for aquaculture feed production. Then of course photobioreactors are used for production of high value products, because when it is about the nutraceuticals we need to maintain the quality of the product. So the factors that affecting the microalgal cultivation are light is very much important for photosynthesis. Simultaneously adequate temperature plays an important role. Then medium compositions, pH, the type of algae, of course it would be very much critical, because if the algae is not potential or it is having a very low growth rate, it will affect to a large extent as well as to the commercial angle as well. Aeration, of course, because in any of the microalgal growth process it takes CO2 or air from the atmosphere for its growth. And of course mixing, because through the mixing you can just maintain the cells in a very uniform conditions, and of course photoperiod is as well as important. So coming into the commercial angle, The reason why we select microalgae is that it provides a variety of products apart from rather than a single product. For example, if we are going with any of the microbial system wherein bacteria or yeast is being produced, only one or two types of products can be produced. But in the case of microalgae, we can see that we can go for a variety of products. So the strategy for domestication to the market means starting from scratch to going to the market. The various steps that are involved is firstly is the strain selection. We have to identify a potential strain that can produce commercially important products like nutraceuticals, antioxidants products, pigments, omega-3 fatty acids. Then also there are various products which are being used in animal feed, aquaculture. Simultaneously, biofuel is also one of the area, and biochar also can be produced from the residual biomass. So after the strain selections, we need to identify the products, and after the identification of the product, then we need to identify a proper scale up strategy for taking it into the pilot scale and going for the commercialization. So in the microalgal cultivation strategies, also apart from various types of reactor systems, there are three different types of process that are being employed. One is continuous, other is semi-continuous process, and batch process. Batch process is very easy, and it is very easy to handle as well, but as far as the commercial angle is known, most of the processes are either semi-continuous process or continuous process. These are the pictures of various photobioreactor systems as well as of the raceway ponds. So the strategy, or we can say the process flow chart that we employ for mass cultivation of microalgae comes from the inoculum. Firstly, from the lab wherein we have let's say 100 ml or 1 liter of thick seed culture needs to be further scaled up into let's say 1000 liter or 1 meter cube pond, which would be taken into the another the pond, which will also be served as the seed inoculum for the mass cultivation of the microalgae. So in the mass cultivation means for the pilot cultivation of the microalgae, we may use CO2 or seawater can be used as a nutrient media. In some cases, fresh water with other nutrients are being used as well for the nutraceutical purpose, or where availability of seawater is not there. So during the cultivation also we can see that there would be, when we are cultivating in open raceway ponds, there would be certain level of evaporation would be happened. So in that case we have to top up with the fresh water or with sea water or media components to have a means completely balance of the nutrients for having a good productivity. So after the mass cultivation is over, means after the cycle of the cultivation of microalgae is complete, let's say 15 to 20 days, sometimes it goes from 10 to 12 days as well. So after the cycle is completed, then we go for the harvesting of the cultures through either centrifugation or by adding any flocculant or via through membrane system. So we take the cell mass out of it, and the residual water are again recycled into the cultivation system to have a means better economics, because the cost of water adds means quite a more percentage in the costing. So anyway we have to recycle the water that is left over to have a cost effective process. So after we get the microalgal biomass, we extract various different types of products, starting from the lipids, then various value added products like carotenoids, and pigments are there like phycocyanin and other products as well. So after we extract all the products, then the residual biomass can either go as an energy feedstock for the pyrolysis or for biogas or for biochar. any of the process can be used to valorize this residual biomass. So coming into the cost economics, so when we plan for any process at a pilot scale, we need to first figure out a basic cost of the production to have a complete picture that whether we are having a sustainable process or not, whether we would be gaining return on investment on the investment that is being put in our process or not. Means we need to have a basic cost benefit analysis at the initial scale during the pilot experiments to have a complete picture on the sustainability angle. So the basic things that are needs to be considered during the cost estimation are the capex and opex. Means in the capital equipment size and type of equipment needs to be measured. Then what are the total lifetime and taxes for the depreciation needs to be measured, which would be part of a the capex cost. Means for example, whenever we are going with any of the process, let's say we are thinking of producing astaxanthin from microalgae. So after having a complete mass cultivation and downstream strategy, we need to have a proper planning of the capex and opex. Means capex means what are the various capital equipments, like we will be having raceway ponds for the mass cultivation, there would be certain pumps for pumping out the culture, pumping out the seawater, etc. Then there will be shade for storage of the biomass. Then for the downstream also there will be filtration units or centrifuges or any other equipment would be there. So whenever any equipment is working, at least for 8 hours at a day, there would be certain depreciation cost. So we need to calculate the depreciation cost as well when we are doing the costing, and then simultaneously from when we are operating at a batch scale or a semi-continuous scale. So every day there would be certain raw materials would be used, certain utilities would be used like steam, heat, whatever the utilities that are being used on daily basis, and of course manpower like labor and for the supervision as well, the manpower being involved. So based on this we will be having a complete figure of the production cost, and based on the production cost we can just have a complete picture on the and have a comparison of other process, means where do we stand. So based on that we can just think it of whether we should go to the market or we still needs any optimization. So this is a basic model just taken from the European context. Then comes I would like to discuss whatever the various microalgal research currently we are working on. One of the area for us is the biofuel, wherein we are still doing the microalgal cultivation in our salt farm for biodiesel production. Then pigments like phycocyanine, phycoerythrin, carotenoids, and of course value added products like PHA, biofertilizers and other products are being extracted from the residual biomass. And also bioremediation is one of the technique, because as I was telling previously, the cost of water adds huge to the basic costing. So I think this I have discussed on several domains as well. So we were working on a CSR Nimetli project wherein 9 various partners across the globe were involved, wherein the job was to identify a potential strain that would be having a maximum lipid content and biomass productivity. And as an outcome of this, initially we have went directly onto nature, and from the nature we found a microalgal mat then that can be skimmed off from the environment, and lipids can be extracted from it, and using this we have run an unmodified diesel vehicle using B20 biodiesel. So the journey doesn't end there. So after that we were into the another strategy wherein we are doing the mass cultivation of Chlorella variabilis for generating the sufficient biomass. So in this case we were able to identify a potential microalgal strain that is auto settling in nature, means we don't need any centrifuge or any such process, just to dewater the thing. Means the after 12 days I think the microalgal cells were settling down, and we can just transfer that culture to another pond and take the biomass. Of course, after there were certain different issues comes into picture. Likewise, every year or every season we might have noticed the environment fluctuation is huge. So whatever the temperature that is now, in the next season it would not be there. So in this process also it was noticed that when we were working on the microalgal cultivation during 2012 2013, scenarios have changed. 2014 again scenarios have changed, and now also we can see the scenario is totally different as per as the temperature is concerned. So in this case, at the current context, we have to just dewater, means use the centrifuge for the dewatering purpose. So this was the heat that was prepared. So after getting the desired biomass, we have to extract the total oil out of it. From the total oil or the non-polar lipids that was obtained, so we have prepared sufficient amount of biodiesel, which was used for again running an unmodified diesel vehicle, and this time we have run the vehicle with B100s, means the neat biodiesel. This was the POC that was cleared. So after that, we have sent our biodiesel for engine testing at IIP Dehradun, and the IIP has reported that the carbon emissions were in comparable range. So you can see, apart from diesel, it is less, and the oxidation stability was also found to be means quite good. These were the various media reports wherein the work was highlighted. So basically, in the whole process, the challenges that we found were the environmental fluctuations during mass cultivation. Of course, the airborne contamination under mixotropic mode that needs to be tackled. Energy output to input ratio with the conventional oil extraction was less than 1. Seasonal variation of lipid content in large scale open cultivation is still a big challenge, which we are trying to overcome with various synthetic biology approaches as well. And obviously, preserving the inoculum is one important step during the off season, means where during the season wherein it's rainy days, so we need to preserve our inoculum so that it doesn't get lost. So the most critical things we found during the whole process was natural harvesting is possible. We were able to harvest autosettle biomass from a very potential species of Chlorella variabilis, which was isolated from west coast of India. We have identified best strain of Chlorella, which was halotolerant and thermotolerant as well. Then some seawater was utilized for the cultivations. So also we have utilized solar energy for biomass production as well. So coming into the benefits of the process, if we think of the economic angle, we all know that all the prices are fluctuating, and in the current scenario, it's quite fluctuating to a very large extent. Secondly, we have utilized solar energy, which is a low cost technology, and as far as the environmental benefits are concerned, it can reduce carbon dioxide, and also nowadays we are planning to use flue gas for the microalgal cultivation. Of course, coming to the social benefits, we are working in an area wherein we are utilizing the unutilized land of salt farms. So obviously it would be giving livelihood to the salt farm workers, which are known as agarias, and it will also lead to the women endowment. This was the picture wherein solar based extraction was tried. Okay, coming into the valorization of the residual biomass. Residual biomass can be directly used as an biofertilizer applications, because it is having a certain good concentration of nitrogen which can replace the chemical fertilizer. So again, after this process, we notice that we need to optimize the process wherein we need to use the CO2 present in the flue gases for microalgal growth. So in one of the project we have collaborated with Adani Power Mundra, which is known as one of the biggest power based company. So you can see the picture of Adani Power Mundra, which is located in the Kutch. It is the largest power plant in Gujarat as well as in the globe. So we have tried to just capture the flue gas, wherein 15% CO2 is present in the flue gases, and that can be captured for the microalgal growth. However, the optimization and other various process optimization are still going on, and maybe I think in couple of years we would be coming into a sustainable process. But till date the process is still being optimizing, and we are trying to get good biomass yield using the, because the problem is that using flue gases or using CO2, microalgae can capture at a certain level only. So in that case, nowadays we are also exploiting the various synthetic biology approaches wherein we can develop certain strains which can tolerate the CO2, let's say more than 5% or more than 10%. So this is the basic process design flow sheet. This is also means the PID diagram of our process. So again comes key, after having a complete process also there would be certain byproducts present in it. So likewise, after the biodiesel is produced, there would be crude glycerol present in it. So you can just see key algae biodiesel waste residue means the crude glycerol can be utilized for preparing polyhydroxy alkanoids, which is a well known bioplastic. So we have developed a process wherein crude glycerol and the hydrolysate of the residual biomass can be prepared for preparing polyhydroxy alkanoids as well. And there were some other process wherein levulinic acid from seaweed can also be utilized as a precursor for improving the quality of the pH. Means utilizing levulinic acid as well, we can improve the properties of pH for the packaging sector, because we all know that the PHA is having a very less strength. This was the bioplastic film that was prepared. Even we have tried extracting of PHAs using ionic liquids in collaboration with our natural products group. So then comes another approaches, means when we are working with the microalgal based processes, as I was telling previously, the cost of the water or seawater adds certain high percentage in the costing scenario. Means for example, the cost of seawater or freshwater is quite huge as well, and lot of water is required in the microalgal process, and some concentration of water also gets wasted. So in that case, so we were working on a bioremediation process using for bioremediation of textile waste water, but somehow it has lead to development of certain nutraceuticals as a breakthrough. So here you can see that the textile waste water, which was having 60% of CO2, COD. So this, sorry, it is having, I think, more than a lakh of COD present in it, and we were able to reduce 60% of COD, and having a treatment cost which comes around, I think, 2.43 rupees per meter cube. So, so in the whole process, what we have done, we have used the textile waste water for the growth of microalgae without any addition of nutrients, because textile wastewater is already having sufficient good concentration of bicarbonate salts and other salts which are being used by the microalgae for its growth. Basically, the nutrients that is required for the microalgal growth are present in the textile wastewater. So we have used the textile wastewater for growing the microalgae, and you can see that the at the 6 days the color got completely degraded. Means at the one instance we are just degrading the dyes as well as the nutrients that is present in it being utilized by the microalgae for getting a sufficient good biomass. And if we think of the economics as well, it is on a comparable range to the commercial processes. So what we are doing in this process, when we are growing the microalgae using the textile wastewater, it would be a mixotrophic process because the ammonium bicarbonate and other organic salts are present in it, which are leading to a mixotrophic growth, that is heterotrophic as well as phototrophic. So in this case, we are getting the biomass yield of, I think, let's say 6 gram per liter within 6 days. So that is a breakthrough, we can say. And from this microalgal biomass, we were able to extract total lipids. From the total lipids, initially we are extracting the gamma-linolenic acid, which is a well-known nutraceutical, and it is being used for treating the premenstrual syndrome in women. So, so after extraction of this total lipids, we are purifying this gamma-linolenic acid, and then the residual oil process further for the non-polar lipids, and biodiesel can be prepared from the non-polar lipids. So after this also, the residual biomass that is remained is processed for the enzymatic hydrolysis, and using this enzymatic hydrolysis we are producing biopolymers, for example epsilon polylysine. Even we can produce PHA also from the residual biomass. So this is the complete material balance, where you can see that we are not leaving a complete even a single thing in the whole process. It is a complete zero waste process, wherein after extraction of the lipids also, lipids we are extracting GLA, means gamma-linolenic acid, and then from the residual biomass also we are extracting the protein rich fraction, which is which can already go for a vegan supplement, and the residual biomass can be processed for the enzymatic pretreatment followed by microbial fermentation for having a polylysine, which is a well-known natural food preservative. So coming into the hydrolysis angle, apart from enzymatic hydrolysis, for the first time we have reported the hydrolysis using detergent, that is sodium rhodioxide sulfate, which is a well-known detergent. So we were using detergent for the hydrolysis of the residual biomass, and which lead us to obtain sufficient good concentration of reducing sugars from it, which was processed for the fermentation. This was the medium and upstream conditions. So using this we were able to produce polylysine, and for extraction and purification also we have utilized ethyl ammonium nitrate, which is a ionic liquid, for a single step purification. You can see this is the process wherein this may be one step or two step purification, wherein the total proteins are first precipitated using ammonium sulfate, and then using ionic liquid we were able to precipitate the total amount of polylysine. And if we compare this with the conventional techniques uses at least 5 to 6 different processes, starting from ion exchange, gel permeation, etc., etc. Okay, this is the basic flow sheet for our gamma-linolenic acid production at the pilot scale. So you can just see the products out of it. This is the basic material balance. Coming into the production cost, using the CSM-CRS developed process, we are able to get this product at 890 rupees per liter, and if we compare with the market cost goes from 2000 to 3000 per kg. Okay, another process wherein we have used fishery wastewater as a nutrient media for the growth of microalgae. So, using fishery wastewater, we were able to get various products from the microalgae, and one of the important nutraceutical products was astaxanthin. So, utilizing this fishery wastewater, we have grown identified strain of Chlorella vulgaris in open system, and after for 12 days the biomass was harvested, and this harvested biomass was initially processed for total lipids. So from total lipids we have extracted the pigments, that is astaxanthin, and then again the non-polar fraction was processed for the biodiesel production, and the biodiesel waste residues can be utilized for the biodiesel production, and the residual biomass can be process further for the hydrolysis, followed by the fermentation of the hydrolyzate for getting the biopolymers, for example, epsilon polylysine. We have also calculated the E factor, and it was also showing the green process. So cost of astaxanthin is at the commercial scale is known to be 12,000 per kg, and our process is still at a bench scale, we can say, and we are just means trying to scale it up at a pilot scale for getting the product. Okay, we all know a very important product from spirulina biomass is the phycocyanin. So we have established protocol for extraction and purification of phycocyanin as well. But recently we are also working on a single step purification of phycocyanin, wherein we have some leads. Likewise, we are using a designed membrane which can just purify the phycocyanin at a purity of, let's say, 99.9% purity using single strains. Means what we are doing, we are just cultivating the spirulina biomass. From the spirulina biomass, we are extracting the total proteins, wherein phycocyanin is coming in the ammonium sulfate, means we are processing the protein precipitation using ammonium sulfate, and the extract is coming into the buffer which is going for the dialysis, and at the same time purification using our design membrane. So another area is when we were working on this process. So we on the process of the biopolymers production or bioplastic. So we found that in order to reduce the cost of these products, why not to have a simultaneous production? Because we noticed that one of our bacterial strains is able to produce both epsilon and polyhydroxyalkanoids. So the strategy that we employed is that after the fermentation, we are extracting polyhydroxyalkanoid as an intracellular product, and the extracellular product polylysine to be extracted from the same fermentation broth. So these are the nutrient upstream process conditions, like initially the seed culture is growing in the nutrient broth, followed by production medium wherein crude glycerol is as the carbon source along with other critical nutrients is being added, having certain pH temperature, and the polylysine yield at a scale of 0.2 gram per liter is obtained, wherein the PHA yield at a concentration of around 65% with respect to cell derivative is obtained. So coming into the downstream process, again means we are doing the purification of epsilon polylysine using in a two-step purification, wherein initially proteins is being taken out using the ammonium sulfate precipitation, followed by dialysis, and the precipitation was done using tetraphenyl borate anion. This is the NMR of the structure of the polylysine, and the type of PHA that was obtained was polyhydroxy butyrate copolymer with the valerate, and these are the TGA and TSC. So this is our small research group, and we were having collaborations with the various other institutes and across the globe as well. So we are having collaborations with National Research Center, Cairo, Egypt, then Sapienza, then with the Nebraska as well, and industrial collaborations with Centev, and in India also J Oil, Adani, and Edo Biotechs are there. So that's all. I think if any questions are there, I would be happy to answer them.
Yeah, Sorish. Yeah, Dr. Sorish, thank you very much for your interesting talk. And I, Dr. Anupama Srivastav, on behalf of our Life Sciences Department, Parul Institute of Applied Sciences, Parul University, would like to thank you for sparing your valuable time and knowledge for our students. And I'm sure our students might have imbibed a lot of knowledge and might have generated a lot of interest towards the research area now. So thank you very much, and we are looking forward for many other few collaborations to be done with the CSIR, CSMCRI in future, and with you also. Yeah, thank you.
Yeah, thank you, ma'am. Regarding collaboration, anyone is welcome to come to CSMCRI and work with us.
Yeah, sure. Yeah, we will motivate our students to join CSMCRI for research. Yeah. Thank you.
Okay, yeah, thank you.